subphylum Mandibulata, which was kind of vague what that all meant. What it really means is the mandible has 2 hinges rather than 1 hinge. But you can't see it, it's kind of hard to describe. So in the last 20 years, there's been a push to change the name of this phylum, which includes the crustaceans and the hexapoda, into what's called the pan crustacea. Pan meaning, including all of them. Pan, like, pan meaning all inclusive. And the crustacea kind of implying that the stem organism, or the common ancestor of this, was a crustacean-like organism. So last time we covered the crayfish, in addition to things like barnacles and isopods and crabs. And hopefully you were impressed at the diversity and anatomy of the crustacean. But the class Hexapoda is actually even more diverse. So here we're talking about the most diverse group of organisms, a group of animals, certainly, probably including all of the organisms as well. But it's just incredible diversity. For some reason, the Hexapoda, which includes the insects, have just taken off, and they've dominated the diversity of animal diversity in the Paleozoic, from the Carboniferous on, in the Mesozoic during the time of dinosaurs, and the Cenozoic. This group, the Hexapoda, just seems to keep coming back with more and more variation and diversity. So let's go ahead. I'm not going to redraw the whole list of classification like last time. We're going to focus just on this group called the class Hexapoda. Now, as opposed to the crustaceans, the kind of things the Hexapoda have that the crustaceans don't have is they have 1 pair of antennae. If you remember, the crustaceans had 2 pairs. There was the first pair and second pair of antennae was kind of the common thing. Crustaceans, all the hexapods only have 1 pair. The name Hexapoda, hexa meaning 6 and poda meaning feet, we have 3 pairs of thoracic legs. And typically, the abdomen has no, or if they do have legs, very few legs. I'll give you some examples of where abdominal legs happen in hexapods, but nothing like what we see in the crustaceans, where it's like legs, 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 and more legs, and then after that, even more legs, right? It was just like the head had 5 pairs of legs, and the thorax had, 8 pairs of legs. The abdomen had at least 8 pairs of legs. So crustaceans were just kind of all about legs. The hexapoda really is just 3 pairs of legs that kind of made it efficient down to the thorax region. And that's kind of their thing. Then we have the evolution probably in the late Devonian, certainly by the Carboniferous. Basically wings appeared. And I'll talk about what the origin of the wings are, but wings basically essentially gave this organism kind of like the ability to disperse, occupy new habitats, and incredible adaptation to flight. And so what the definition of an insect is a hexapod that has wings, or at least has evidence that their ancestors had wings. So the term insecta, or insects, refers specifically to winged hexapods. But not all hexapods actually are winged. So let's go ahead and first off, let's take a look at kind of what makes a typical hexapod in terms of its body parts. They have basically 3 body taggings. There is a head. There is a, I can't remember what color I used before. We'll roll with it here. There's a head, a thorax, and an abdomen. So they have three typical body tagments. So we have an abdomen, we have the thorax, and we have a head. So I guess the main difference is compared to the crustaceans is we called this a cephalothorax crustaceans. It was a combination of the head and thorax as one segment, one body tagment. This one, the head tends to be separated from the thorax, so it's a separate view. Let's go ahead and take a look at appendages. We have one pair of antennae. The antennae are almost always sensory. So antennae. We have one pair of mandibles. The crustaceans and the Myriapoda had two pairs of maxillae. I usually listed them as first and second maxillae. Insects have basically the same setup, except the second maxillae are fused. And you know, I'm giving you more information you need to know. I'm not going to be able to ask you about this on an exam because they're kind of too small. But we basically have the maxillae, and then the second maxillae is actually modified as a lower lip called a labium. I can't really ask you that on a test because it's so hard to pin and identify. It's kind of really specific to entomology. But just note that this is really the second maxillae. But it's just modified into a fused lower lip. So insects kind of have this mouth part that kind of has like a package. There's a lower lip and kind of an upper lip above the mandible called a labyrinth. And so they have these kind of more condensed, really specialized mouth parts. On Thursday, we're going to go over insect diversity. I'm going to go over like 15 different orders of insects for you. And I'll show you that some of them, for example, like mosquitoes, have modified their mouth parts into a stabbing, blood-sucking mouth part. Butterflies have modified it into like a soda straw, coily mouth part. Houseflies have modified it to a spongy mouth part. Super flexible adaptations in terms of mouth parts. But you can see it's just really efficient. Just a few parts that are reported. Okay, and then for thoracic legs, the typical system is to have the legs for walking. But on Thursday, I'll show that we have some adaptations, for example, like jumping. Grasshoppers have a jumping leg, mantises have a grasping leg, cockroaches have a running leg, there's digging legs. So the legs can be modified for all kinds of functions here. I'm just going to just draw a generic leg here. But here we have our walking legs. And then, as I suggested, the abdomen normally doesn't have any appendages, and if they do, it's a circe-like appendage. Some orders of insects have circe, many don't. So it's kind of like a optional feature for many insects there. Okay, one key feature, though, that we can't ignore is this whole notion of wings. The wings are extensions from the second and third thoracic segment. So these wings, if they're present, that's what makes a true insect, or at least vestigial remnants that wings had existed. The wings, there's two pairs of them primitively, and they occur on the second and third thoracic segment. There had been a lot of debate as to what the origin or evolution of wings are, but I think the consensus now is it's a modified gill, that the structure of wings and where they originate from is very, very similar to gill structures that occur in aquatic insects. So it's likely an aquatic type insect that had a gill on their body, on their thorax and abdomen, when they emerged into an adult, retained that gill-like feature, probably or possibly, when no one was there, but possibly used that structure to glide, like get up onto high structures and jump off and then glide, and then eventually evolve it into a flight mechanism. But more recently with DNA sequencing and DNA technology, they've actually shown that the genes that code for the gills in aquatic insects are pretty similar or the same as the genes that code for wings in insects. So it's pretty clear that this is a modified gill. So it's a great example of an exaptation. Take a respiratory structure and then, essentially modify it so you can fly with it. It's pretty outrageous kind of to think about, but it seems to be the consistent pattern of what the structure of gills of wings are. Okay, so huge diversity. We're going to go over insect diversity on Thursday, but I want to touch on this idea of what's the difference between a hexapoda versus an insecta. The ones on the left, the three on the left, are what I call non-insect hexapods. And you've probably seen this, silverfish and jumping bristletails. In fact, these are really common now. If you find these jumping bristletails or silverfish, bring them in. They're really cool. I actually have I think I have a jumping bristletail in it's preserved, but it's in a dish over here. But it's hard to see here, but this organism here, which is called a jumping bristletail, is a common name. It actually, you can kind of think of it as like a link between crustaceans and insects. It has 3 pairs of legs, it has 1 pair of antennae, but it actually has abdominal appendages that kind of resemble swimmerets. They don't use them for respiration or holding heads, they use them to help them jump, but they actually have abdominal appendages on every segment, like a crayfish does, or like an isopod does. And then they have, seriously, this third filament is a terminal filament that's basically an extension of its gonads. But essentially, it's kind of like a shrimp on land. But ultimately, it has features of a crustacean and features of an insect kind of combined in the same organism. So this is considered a very, very primitive hexapod that probably is a good demonstration of why the hexapoda and the crustacea are kind of combined together in this group. This is called a collembola. They're kind of soil hexapods. But the main thing to note with those hexapods is they have no evidence for any structure that resembles they ever had wings at all. Whereas we have 2 of the more primitive type of insects. Probably when insects first developed flight, they probably resembled what we consider like a mayfly. And if you're into fly fishing or angling, you know that mayflies are aquatic insects that when they emerge, they use their wings to disperse. But mayflies are kind of clumsy flyers. They're kind of like, they look really awkward. They're kind of using more air currents to drift around than to really power their flight. But then we have like dragonflies and damselflies, which are like super amazing, powerful flyers. Dragonflies, are able to kind of zoom around like a little jet plane, monitoring their territory and catching prey. But these are examples of insects that exist today that probably resemble the insects that were diversifying in the Carboniferous. So mayflies and dragonflies. We'll talk about them on Thursday. Okay, so I think given that, what we'll do is we'll look at our cockroach kind of as a model insect. So today is going to be on morphology and anatomy. And then Thursday, we're going to focus in on looking at about 15 different orders of insects. And I'm going to actually have you develop a dichotomous identification key, kind of like a cheat sheet that you can use on the exam to identify orders of insects, common orders of insects. And I think that'll be. Well, let's look at the cockroach. We have these are called Dubai roaches, and they're sold at feed reptile stores to feed things like lizards, and mostly lizards and geckos like these. And so I'd encourage, if you want to, these are nice cockroaches as compared to the cockroaches that you find in restaurants in town. These are not super fast runners, so they're kind of easy to catch. So I have a few left over here, so you can actually handle these pretty well. And you know, they're pretty cute. I mean, so anyway, you know, if this was a German roach or an American roach, it would be like out the door, like by now, right? But these are pretty docile, and you know, kind of have a little personality. But anyway, these because they're so fatty, are useful for feeding like geckos and things like that. But anyway, this one doesn't have any wings. It's an immature. So in the immature stages, when they go from an egg through their first few molts, if they lack wings, they're not sexually mature. You know, let's go back. Go away. Okay. And but in here, there's one remaining that has wings. Fortunately, these don't fly. This species doesn't fly in this climate. But actually, I just saw my first, just this last couple of years in Reading, flying cockroaches. I didn't think California had cockroaches that flew. And you know, great. So anyway, so this one has wings on it, and on the ones that I've euthanized and dissect, I think all of them have wings. So I'm wanting to kind of see where the wings are, what they are. But when they have wings, basically, that's the adult stage, and they'll never get any larger. They basically molt, they shed their skin, their exoskeleton, up to the point where they sprout wings. And so you can kind of see the wings are kind of like, you know, I'm like cellophane or like kind of plasticky. And really for the cockroach, the wings are kind of more protection than anything else. Okay. Anyway, so feel free to handle those if you want to. I've euthanized them in ethyl acetate, which is like nail polish remover. And what happens, a couple of interesting anatomical features I'll talk about with these is their respiratory system is totally different than any other organism. The insect's respiratory system is a unique to insects or hexapods kind of thing. So, for the cockroach, what you're going to do is you're going to, even though the diagram here shows the opposite of what I'm saying, I want you to dissect them from the dorsal side. Dissect them from the dorsal side. So here we go. I'm going to go ahead and draw the cockroach here. Okay. So what you're going to see basically is the head of the cockroach typically is kind of tucked underneath. Their head is kind of tucked back, and their mouth parts are kind of pointing backwards towards their belly, or towards their ventral side. So that's kind of why I'm kind of drawing this where you can barely see the head. This is the first thoracic segment called the pronotum, and it's kind of like a shield over the first thorax. The wings are basically here. But what I'd say is the wings look like this. They're kind of folded like this and then folded over the body. What I'd say is pull the wings out and then use scissors to cut the wings off at the base. And so when you do that, when you cut the wings off at the base here, you're going to end up with kind of like these little cut off stubs here. But those are on the second and third thoracic segments here. You can see something like this. The second and third, first, second, and third thoracic segments like that. Then the abdominal segments are essentially, they look the same. There's just no legs attached to them. And the abdominal segments have these plates, these articulating plates that go back. I think there's probably 8 exposed abdominal segments. I'm just going to draw like 5 of them to get the idea before you get the terminal segment here. These typically have cerci, so you'll see a couple cerci sticking out here. And that's kind of the idea there. So there's our dorsal side of this insect. You'll see that we have our little feet here. And I'll I'll draw a more detailed leg here in a second, but typically they look kind of like this, you know, the legs kind of, front ones go forward and the back ones go back. But you take a look at the legs. I'm not going to ask you anything about the leg parts, but on Thursday when we look at the diversity of insects, how they've modified the legs kind of is unique to like mantises versus grasshoppers, etc. So that's what you got going on. Okay, so I think the best way to dissect these is to use scissors. And we have these, if you go over here and dig around in our scissor bin, you'll find these really, really fine point, like, you know, super delicate sharp point scissors. So grab a pair of these, you know, these scissors. You may have to clean them off. Some students have stabbed these into the wax pans, and they get wax on the end of them. So that prevents them from cutting as well. But anyway, use these to do dissection. Scalpels are useless. Scalpels are just not sharp enough and not accurate enough. So we'll go ahead and use scissors to dissect these. And what you're going to do is you're going to cut out like a hatch on the back. So take your scissors and stick the point of the scissor like underneath the plate here on the abdomen, you know, pointing anteriorly to the head. And then you're going to basically cut like a hatch or a door, probably up to the third thoracic segment, all the way back to the last segment. So essentially you're going to go ahead and cut around the whole thing. Then take some tweezers and grab this bottom of the, you know, where you cut, and you're going to peel it like a sardine. If you're eating sardines or kipper snacks, you know, you kind of crack those open, those little tins, and you peel them back, or like a can of tuna or something. You're going to basically peel it open, peel this forward, so you'll have to hold the tip of the abdomen or pin it down, and then peel this forward, and you want to basically remove this whole hatch. When you take the hatch off, don't discard it. I want you to take a look at what's inside the hatch. If you look inside the hatch, so here's kind of like what you just cut off like this, the hatch, back here. It's actually really cool. There is a combination of the circulatory system and the trachea. So the circulatory system basically is, I'll do it in red, even though they have, okay, they have hemolymph, but they don't have any circulatory pigments. There's no hemocyanin. So I'm just going to draw it red because that kind of resembles a heart color. Each segment has its own heart pump. So what you'll see basically on the dorsal side of this hatch is you'll see each segment has like a little swollen pump. And these are what are called pericardium. They're just kind of inline pumps that go down on the dorsal side. So they have a dorsal heart, basically. And then a key thing I want you to see is there's these tubes, like air tubes, running throughout the system here. And these are called the trachea. So one interesting thing here is the respiratory passageways of an insect are air tubes. And the trachea basically open up as holes inside of the abdomen and the last two thoracic segments. These are called spiracles. These openings here. And they are openings that lead to trachea. So the insects actually breathe through holes in their abdomen and thorax. They don't breathe through their mouth or gills. They have a totally unique system. I guess it's kind of similar to like what we saw in the scorpion, right? The bookworms, but there's no gill structure in here. What happens is these trachea are air tubes that get carried all the way down to the individual tissues So atmospheric oxygen is carried all the way down to the muscles as a gas. The respiratory oxygen never dissolves into a blood of any kind. It's delivered right to the tissues that need it. And for lecture exam 3, I'll explain why this is cool, and that is because our atmosphere has about a 21% oxygen content. You can deliver that full 21% oxygen content right to the muscles that need it, versus dissolving the oxygen in the blood, having your blood cells carry it around and deliver it to tissues. It takes time to do that. That's why when you do exercise physiology, you measure your VO2 max, which measures your efficiency of delivering oxygen to your cells. Insects have an amazingly incredible VO2 max. They deliver oxygen right to the muscle cells that need it as an atmospheric gas. And so that's probably one reason why flight is so common in insects, is even though flight requires a lot of metabolic energy, they're able to basically deliver oxygen right to their flight muscles as gas oxygen and never using blood as a transport. So, for sure, take a look at the trachea in that back half. You'll see trachea all throughout, but I think it's easiest to see it in that kind of orbital. Okay, so I'm going to go ahead and erase this patch that we removed and show you what we're looking at as internal anatomy. And of course, as always, I'm trying to tell you exactly what I'm going to ask on the test. So make sure you see those structures. So let's go ahead and take a look at the digestive system. The mouth opens up into a pharynx, and the pharynx into an esophagus, and so those structures are here in the thorax. You're not going to see the esophagus. What you will see, though, is the esophagus will merge into a crop. So their stomach, per se, is called a crop. It's kind of like what we saw in the annelid worm. They had a crop. These also then have, and you may or may not see this if you're really into it and you have a good magnifying phone or you want to use a microscope, you can see this structure, but it's kind of like a gizzard, but it's called a proventriculus. It's not truly a gizzard because it's not using sand grains or minerals to grind the food. I guess what it's most similar to would be like the cardiac stomach of a crayfish. The crayfish had essentially exoskeleton plates that they used to kind of crush their food as it was going through. But in the crayfish, it was kind of reversed, right? We had the gastric mill, and then we had the pyloric stomach. This is kind of backwards. We have the crop, which is kind of like the digestive stomach, and then we have the proventriculus. Inside the proventriculus, if you look real carefully, there are actually exoskeleton teeth in there. But this is so small, I would not ask the proventriculus on a test because I just can't put a pin in it. It's actually smaller than a pin. But see if you can see that. It's kind of cool. I just wanted to tell you about it. Okay, proventriculus then leads to ceci, or digestive cecum. Ceci is plural for the side pockets that occur for digestion. So ceci secrete enzymes and store microbes like protists and bacteria that help them digest their food. These you will see, and I really like to pin these and ask them on the test. So the ceci are pretty obvious, and you can see those pretty well hanging out. Okay, then there is a long stretch of what we're going to call midgut. The midgut is the absorptive region of the intestine. So the midgut, what it's called. The midgut will be covered in trachea. So you're going to see trachea all over the midgut. So be sure you see that. This is trachea. And they're basically supplying oxygen to digestive cells in the gut. Okay? All right. Then the hind gut is sometimes called the colon. The gut will basically enlarge or swell here, and so this is the hind gut. Or I guess sometimes it's called the colon, but the hind gut is the more accurate term. All right. So that basically is the digestive system. There is one more structure I really want you to see, and in fact, I will do my best to label it. It'll probably be identified on the test. And that is that there are these thread-like structures that branch off at the junction of the midgut and the hindgut. And these are totally cool, unique osmoregulatory and nitrogenous waste excreting structures. And these are called Malpighian tubules. Malpighian tubules are the unique to arthropods, terrestrial arthropods. They're found in insects, spiders, and scorpions. And they're found in myriapods as well. These are how they collect their nitrogenous waste and excrete it as a uric acid. So for lecture exam 3, we'll go over how these work, how they function. But what they do is they're kind of like if you or you know someone that has a swimming pool that has they have like that hose that kind of whips around in the bottom of the swimming pool. It kind of, I think it uses water pressure to kind of dislodge the algae. But in the pool, basically, you see this hose snaking around to clean the pool. These Malpighian tubules kind of lay in the hemocoel, and they snake around. They actually have muscle on them. They snake around, and they absorb uric acid, water, and salt ions. And then in the hindgut, they will reclaim the water and salt as needed, but concentrate the uric acid as a crystal, such that when the insect excretes its waste, the uric acid comes out like a salt or like a crystal, along with the very, very dry feces. And that enables insects to live like super dry environments like the desert. You don't even have to drink water. It's pretty amazing system. Anyway, probably very similar to the excretory tubes in nematodes. Remember that arthropods don't have ciliated cells, so they don't have the ability to have enough nephridia. So nephridia are not an option, but these are super efficient at extracting uric acid from the hemocoel and getting rid of uric acid as a waste with no water needed. So I love hitting Malpighian tubules. Hint. I like in the Siki. And then, everything else is kind of optional, like whether I pin the crop or the midgut or the hindgut would be kind of like the third option. But for sure, plan on identifying the Siki and the Malpighian tubules on the test. And then the third one will be a wild card, depending on what I can get them into. All right? Any questions? Yeah. The Siki are like a digestive pocket. I mentioned before that the definition of a cecum versus a stomach, that a cecum is like this is a cecum, and a stomach is like this. That's literally the definition of a stomach versus a cecum, right? So, like your appendix is off of your cecum on your body, whereas your stomach, is like an inline chamber. So these are Siki, and they're basically, the main function is, which I won't ask you on the test, is secreting enzymes and harboring or storing microbes. So like in a termite, the cecum are loaded with retortaminata, like Giardia, like protists. And so that's very common. Okay, so that's the digestive system. A couple other things to note that I probably won't be able to ask on these for a test, but I want you to see that, is there is a ventral nerve cord. And take a look at this because we're going to talk about this probably for lecture exam 4, is the nerve cord in arthropods is ventral. We talked a little bit about it with the squid, with the donut brain, but the nerve cord is ventral and the heart, or the pericardium, is dorsal. So this is a ventral nerve. And there are ganglia, or little mini brains, at every segment. So this is pretty cruel. I mean, that's why I don't like to do this. I don't normally do it. But years and years ago, with the cockroaches, I told the students that basically you can cut the head off of a cockroach, and it will live and crawl around and do its cockroach thing, other than eating and drinking, for like a week. And the reason you can do that is the ganglia in the head, the cerebral ganglia, are basically specialized in sensory and feeding. That's what they do. And vision usually, sight. The ganglia in the thorax are for locomotion, walking around and flying. And the ganglia in the abdomen are for respiration and maintaining and the digestion. So if you cut the head off, the cockroach will still walk around, doesn't know where it's going. It kind of like, it just basically has a mechanical, automated step. So they'll walk. And in fact, in some insects, if you pick them up off the ground and the feet sense there's no more ground, they'll start flapping their wings. So it's like, they're like little computers. All these little segmental ganglia basically contain the program for how to operate that segment or those regions of segments. So there's thoracic ganglia, abdominal ganglia, and cerebral ganglia. So the brain is basically spread out throughout the whole. We don't call it a brain. We call it ganglia because each segment has its own cluster of neurons that manage that segment. But it's interesting to see, this is probably the best organism to see this ventral nerve. And I think some of you saw the brain of the squid, which is cool. It's the same kind of color. It's like a pearly white material, but you'll see these segmental ganglia. And because on lecture exam 3 or lecture exam 4, we'll talk about the evolution of the brain and nervous system, I think it's kind of cool thing to see. But I'm not going to ask you that on test, but I wanted to point it out since we got these cockroaches to check them out. Okay? All right. I think that looks pretty good. If you want to, with your preserved one, or your euthanized one, I'd encourage you to actually cut the head off and take a look at it under magnification of your phone or a microscope, and just look at how neatly packaged their mouth parts are. Pretty fascinating, and that'll help you kind of understand how they've modified these into stabbing and sucking, or spongy, or soda straw. Basically, the mouth part modifications are a major adaptation that happen for insects. All right. So I'm going to go ahead and put one pan out here. The ethyl acetate is a little smelly, so I want to get that kind of vented out. So I'll put this paper towels here. Again, you'll want to get a pan. If you do pin them in a pan, pin them closer to the side or edge in case you want to use a second microscope. But I think so far they actually do a pretty dang good job of getting that going. So we can pull this out. And let's I think I have 8 of these. So some of the bigger tables, if you want to get 2, that's okay. And if we run out and everyone wants 2 per table, I can euthanize more. Let's go ahead and start with these 8. And get your pans. Again, get your very fine tip scissors. And then there's some pins over there. And I'll come around. All right. There you go. It's like Costco free samples. Don't rush.